Hi everyone, I am Rutvi, currently a third year undergraduate biomedical sciences and research student. Today I will be presenting my poster on gene expression meta-analysis of colorectal cancer tumor cells, which reveals genes which are associated in the process of tumorigenesis. So to start with introduction, this is a study in which we wanted to find out that are there any specific gene signatures that are associated with colorectal cancer diseases. So colorectal cancer is very deadly and approximately 12 million people are diagnosed every year with colorectal cancer. So in this experiment or this study, in order to find out the gene signatures, we selected a few data sets that had samples of colorectal cancer versus normal samples, and by using those data sets we elucidated 13 gene signatures. Now the data sets were divided into three types and they were used for the following things. First is gene identification, second is gene verification, and third is gene comparison. For gene identification, we used the two data sets which are mentioned over here in the method section. After using those two data sets, we got a gene signature. Now those gene signature could be set data set A as a reference versus set data set B as a query, and vice versa, that is set data set B as a query versus set data set A as a reference. So by using this, we derived the gene signatures, and whatever genes fell in this category were the gene panels that we used to identify genes in the other data sets. Now these gene panels had 77 genes in the positive panel and 55 genes in the negative panel, and then we compared and verified this among the other random models. Now these random models were used to exactly find out that are these genes only specific in humans or are they also found in other kinds of colorectal cancers like polyp tumors and hyperlepsia and further on. So out of like 55 genes that are present in the positive colorectal cancer gene panel, 54 of them were continuously showed, verified, and present in the gene signatures of other glandular tumors also, whereas 77 genes that were present in the negative gene panel, out of which 75 were constant throughout the other gene signatures that were identified, and they had p-value less than 0.01 and less than 0.05. So this showed that our statistical significance of the shared genes is must, and hence we went ahead and confirmed the gene comparison by comparing it with the random model verification or verifying in other patients that had another kinds of samples. So what do we conclude from our study? So this is usually a bioinformatics study, that is mRNA microarray analysis, which predicts the genes that are potentially involved in developing CRC tumors, examining gene signatures. We found 54 overexpressed genes that most important were solute family carrier genes, calcium genes, and amongst them were the most prevalent solute carrier family genes. Now, these genes were highly overexpressed and shows that their association with tumorigenesis in the process of colorectal cancer. Not only this, but a lack of experimental evidence and dry lab experiences show that how much more work is needed for actually working in the area of colorectal cancer and gene therapy and gene signatures. However, to verify this work, we need certain kinds of wet lab experiments which stand as a proof that the verified genes and the gene signatures are actually standing out in patients of colorectal cancer. Over here in the diagram of the conclusions, you can finally see that what I've concluded, that is out of the 55 genes from the positive CRC panel, 54 of them were constant, which shows how apt and accurate our results was. Similarly, for the negative panel, out of 77 genes, 75 of them were uniform throughout the signature. However, if the desired outcome is prioritized of the potential gene signatures that have been identified in further laboratory or clinical elimination, the GSEA-based approach would be very, very useful. Thank you so much.